Thanks very much, Jeff. So based on the sort of size of companies we've been discussing this morning, so FujiFilm, as you all probably know, is a pretty big company. FujiFilm Diosynth Biotechnologies is actually quite a small, separate business unit, and I'm only going to talk today about FujiFilm Diosynth Biotechnologies, which is actually probably sort of middle-sized company in terms of companies we discussed today. And the first thing I should say, which isn't on any of my slides, and I realised earlier that I should have put it on, is we have absolutely nothing to do with cameras or photography. So that's the first thing you need to know about our company. So what do FujiFilm, I shall try and say FDB to save me mangling the words even. What we do is we are what's called a dedicated biopharmaceutical CDMO business. And that's really the take-home message from what we do. And I'll try and talk you through that because probably some of those words may not actually mean very much to some people here today. So the contract part of it means that all the work we do is contracted for another customer who actually owns the protein molecule we're working on. So we, unlike some of the companies you've heard this morning, we don't actually have any products of ourselves. We're actually developing and manufacturing those for a third party who will actually own that product when it goes onto the market. And what we do is biopharmaceutical development and manufacturing, and in this case 90% of what we do is actually protein pharmaceuticals. So the products we're making are proteins that we used usually as injectable pharmaceuticals for medical conditions. And to differentiate ourselves from some of our competitors who are just what we're called CMOs, actually the development part of what we do is actually a really big part of our organisation, and we sell that as part of our technical package to our customers. And then across the world we actually have 3 sites. We have a site in the UK, and I don't know if people can see clearly on that map. I've tried to point a little arrow to where we're based. So we're based in the northeast of England, not too far from Middlesbrough. We also have sites in North Carolina and Texas as part of the FDB global organisation. But most of what I'm going to talk about for the rest of today is really about our site in the UK and what we can offer career-wise there. So just very briefly talk you through what essentially is our manufacturing process, because that will help you understand some of the sort of technical roles that we have within our organisation. So it's fairly sort of standard stuff for recombinant protein manufacture. So we start off with a recombinant organism. Sometimes we put that together for customers, sometimes they come along with that themselves. Now for biopharmaceuticals, there are 2 main organisms that people are using to manufacture these things. The first one is E. coli, and the second one are mammalian cells, and primarily Chinese hamster ovary cells or CHO cells. So probably about 90% of the products we manufacture at the moment are made in those 2 organisms. So really quite differing ends of the evolutionary spectrum. But a lot of the rest of the process is actually quite similar for both of those organisms. So the next stage of the process is, having got our recombinant organism, we then stick it into either a fermenter or a bioreactor, depending on what sort of cells they are and who's calling it, where we basically grow up the cells to make the product. Out of that we end up with a mix of cells containing the protein we want, but also all the other bits of cells and bits and pieces in there. And essentially, because this is likely to be injected into people, what we need to make is really quite pure protein. So the rest of the process is around about purifying that protein from that soup. So the first stage of the process is what we call separations. That's basically separating the cells from the protein, and the rest of the process is basically protein purification. So it's actually pretty standard protein purification techniques. We use a lot of standard chromatography methods to purify the protein from all the other host cell proteins that are there. And then what we end up with is a protein that's around about 95% pure, or greater than 95% pure, I should say. And we fill that into basically quite big bottles, which is what's called bulk drug product. And then that goes off to somebody who then puts it into vials or whatever final formulation actually gets to the hospital. So reflecting that, these are the functional groups we have in R&D. Now I've deliberately put the R bit of this in brackets because although the group I work in is called research and development, probably 90% of what we do is actually really development. So we don't really do very much actual research. So if you come along and are interested in a job with us, don't get the impression you're going to be doing lots of really cutting-edge research. You're still doing science, and it's going to be still quite interesting, but it's not going to get you lots of nature papers. And the technical groups we have reflect of a process that I've just described already. So we have a group which we call upstream processing, which covers molecular biology, the microbial fermentation, mammalian cell culture, and that primary separation, so basically separating the cells. And most of the time we do that, particularly for bacteria, it's simply centrifugation to separate out the cells from the rest of a product. The next group is downstream processing, which, as I said earlier, is very much about protein purification. And then the other group, so we have a group called analytical development. So we're making quite complex proteins. The regulatory standards we need to be able to inject those in mean we need to understand the protein and be able to characterize it very clearly. So we have a separate group which are basically developing the analytical tools. Often they're quite specific to each particular protein. And then finally there's a group we call process design, and that's concerned with making sure the process is robust and scalable and can actually be used for commercial use. And then if we look at, this is in the UK, our R&D headcount just in general terms. So in our R&D group, greater than 95% of people have actually got a first degree, and around about 40% have got higher degrees than that. And the split in terms of numbers is purification or downstream processing is the biggest group in terms of numbers. And then after that we have, this is a slightly old slide that says expression and cell sciences. That's upstream on the previous slide. So that's the next biggest group, followed by analytical development, and then some smaller groups on top of that. So that gives you an idea of a mix of people we have on site in R&D. Talk a little bit about career paths within our organisation. So we've actually over the past sort of probably 10 years been expanding quite a lot. So we've gone through several recruitment rounds looking to bring people in. And we try and bring people in with a range of experience, from fresh graduates through people with PhDs through to postdocs. And we give suitable amounts of training to people depending on what their experience is, etc. Now, the amount of experience we require for a particular job will depend on the entry level we're bringing in. So obviously we're just looking for people to come in and just work in the labs. They would tend to be graduates, maybe people who are fresh out of a PhD. People with more experience than that, we'd be looking them to initially maybe start out in the labs, but quickly move into sort of a role where they're managing other people, etc. And obviously the experience you have already in your PhD or postdoc needs to have some sort of relevance to what we're doing. We're not looking for people who've run 10,000 litre bioreactors, because we don't recognise that's not something that goes on, but we're looking for people who do have relevant technical experience. And actually, you know, it's a key part of where we can justify offering somebody a postdoc level salary versus, say, a PhD level salary. I've talked so far only about R&D, because people like yourselves who've got a technical background, those are the sort of roles we'd be looking to bring you into the company in the first place. But actually, although we don't have a formal career structure like some of the people we've discussed this morning, actually we found over the years that R&D is actually a really good stepping stone to other careers within our company. So a lot of the people in other functions have a technical background, they've maybe started in R&D, and areas like business development, project management and commercial. A lot of the people in those areas have actually worked in R&D for 2 or 3 years, got a feel for how our business operates, how we work along with customers, and then have been able to make the move internally to those jobs. And actually, although we don't have a formal career route, it's actually something that does happen quite a lot. So what do we look for in terms of people? So the technical knowledge is reasonably key, but actually a lot of what we do is pretty unique and doesn't go on in universities. So we recognise people coming in will always need a certain level of training. One of the things we do when people first start working with us is actually go through about a month or so of relatively intensive training in all sorts of aspects of a business. And then I think it's partly done so that when people haven't seen the lab for a month, they're desperate to get into the lab and do lots of work when they get there. But also important alongside technical knowledge is having the right sort of attitude. So because we're a contract manufacturer, we're working quite intimately with customers. So a lot of what you do, you're having to talk to the customers about, you're having to present data to customers, discuss technical issues with customers. So you need to be able to do that. And customers come in all shapes and sizes, so from people who have a lot of expertise technically in what they're doing to people who maybe are commercial people. So you have to be able to pitch your science to the right sort of people. Although what we do is working in teams. We're always looking for, obviously, for people who've got that sort of enthusiasm, but also because we work in a very regulated environment, concern for standards is also very important. And I think that's my last slide is really just a summary to put up to summarise the business. So across the globe we have 1,000 staff. There's about 500 people work at Billingham. There are a number of commercial products we make, but you won't have heard of any of them unless you've got particular medical conditions. So I'll finish there. It's gone end of slideshow anyway, it's told me. And I've got to stand over if anybody wants to come and talk to me.